Chapter 2, Red Blood Cells. Structure of the red blood cells. These are the biconcave, circular, non-nucleated cell with no mitochondria, ribosomes or Golgi apparatus. Contains large amount of hemoglobin. Blood group antigens are present on the surface of RBC. Protein spectrin, which gives biconcave shape. Now, what is the importance of this biconcave shape? So first one, can absorb more water and prevents the early rupture. Second, easily folded through narrow capillaries. Third, quick exchange of gases. Fourth, increases the surface area. Thickness at periphery is 2 micron. But here the thickness at the center is 1 micron. That is how the biconcave disc shape RBC looks like. Volume 90 cubic micron. Surface area 120 square micron. Lifespan of RBC is 120 days. So here you must need to remember that lifespan of RBC is 120 days, which is destroyed by macrophages in spleen and liver. Those who don't know which are the macrophages, let me clear. A type of granulocyte is a monocyte. Whenever the monocyte is circulated in the blood vessels, the cell itself is known as monocyte. But when this monocyte coming out of the blood vessels and present inside the tissue, that cell is known as macrophages. So we will see later on what is macrophages, but still I already gave you some basic understanding of the macrophages. Now erythropoiesis, most important. Most important for your full question as well as theory viva. Definition: formation of RBC from primitive stem cell. Erythropoiesis is a type of hemopoiesis. Hemopoiesis simply means development and maturation of different types of blood cells. Erythropoiesis means development and maturation of erythrocytes or RBC or red blood cells. The third term is leukopoiesis. Leukopoiesis means development and maturation of WBCs or leukocytes. So here we are seeing erythropoiesis in the chapter of RBC. So definition is development and maturation of erythrocytes or red blood cells. Simply formation of RBC from primitive stem cell or PHSC. PHSC simply means pluripotent hemopoietic stem cell. Site in the intrauterine life up to 3 months in mesodermal yolk sac, 3 to 6 months in liver or spleen. Here the fetal liver and fetal spleen, as we are thinking, as we are looking for the sites in the intrauterine life. After 6 months in bone marrow of all the bones. In adults or flat bones along with ends of femur and humerus, where the red bone marrow are present. So here the stem cell, which is known as pluripotent hemopoietic stem cell. From this stem cell, after the several stages of RBC cell maturation, the red cell formation occurs and it is released into the blood vessels or capillaries, which is present inside the red bone marrow or passes, or the capillaries which passes through the red bone marrow. So here the picture of the all the blood cell formation which is occurs from the pluripotent hemopoietic stem cell, which is also known as PHSC. It's a short form PHSC, and the full form is pluripotent hemopoietic stem cell. So step by step we are going to see every cell, but here we are concentrating on the erythrocyte formation. Major changes during the erythropoiesis. So first of all we are going to identify the whatever the major changes which occurs during the process of erythropoiesis. Decrease in cell size, decrease in nuclear size, which later on disappears, as we already know that the RBCs are non-nucleated cell. Cytoplasm is initially blue but later on becomes red. Hemoglobin appears in the cell cytoplasm. Mitosis is initially present but stops later. So these are the major changes during the erythropoiesis. Now stages of erythropoiesis. First stage, hemocytoblast. So first stage is the hemocytoblast. This hemocytoblast is also known as pluripotent hemopoietic stem cell. Which converted to proerythroblast. So here it's a proerythroblast. Proerythroblast converted to early normoblast. Early normoblast converted to intermediate normoblast. Intermediate normoblast converted to late normoblast. Late normoblast converted to reticulocyte. And finally the reticulocyte converted to the mature RBC. So this is the steps in the formation of mature RBC. So first of all, this pluripotent hemopoietic stem cell PHSC or hemocytoblast converted to proerythroblast. Proerythroblast converted to early normoblast. Early normoblast converted to intermediate normoblast. Intermediate normoblast converted to late normoblast. This late normoblast converted to reticulocyte, and finally the reticulocyte converted to mature RBC, which is a non-nucleated cell. Changes during erythropoiesis. So here we already seen that proerythroblast. First of all, there is a PHSC or pluripotent hemopoietic stem cell. After that, all the cell formation occur in this manner: proerythroblast, early normoblast, intermediate normoblast, late normoblast, reticulocyte. This is the reticulocyte and mature RBC. So one important here in this table, we are going to note the three important points. First one, the cell size is decreasing from 50 to 20 micron to the 7.45 micron. Second point, at the level of late normoblast, this nucleus is disappears. So here the nucleus is disappears at the level of late normoblast, and the third point is hemoglobin appears at the level of intermediate normoblast. So these 3 points you must need to take in consideration during the changes during the procedure or process of erythropoiesis. First one, the cell size becomes decreasing. Second one, the nucleus disappears at the stage of late normoblast. Third one, cytoplasm. Inside the cytoplasm, the hemoglobin appears in the stage of intermediate normoblast. Factors affecting erythropoiesis. Erythropoietin. Erythropoietin is a hormone and it is synthesized from the kidney. or inside the kidney, the erythropoietin synthesis takes place. So normal blood oxygen level. So now we are going to see the which are the stimulating or which are the stimulus for the release of erythropoietin, as well as what erythropoietin will do on the process of erythropoiesis. Whenever the normal blood oxygen level imbalance or imbalance of the normal blood oxygen levels, so the stimulus is hypoxia due to decreased RBC count. So decrease RBC count, so that reduce level of oxygen into the blood. This hypoxia having a stimulating effect over the kidney, so that it release the erythropoietin. Erythropoietin stimulates red bone marrow, enhance erythropoiesis, increased RBC count. So that is how the erythropoietin release from the kidney. It's going to stimulate the red bone marrow, and there is an increase in the release of RBC count, or increase in the release of number of RBCs, so there is increase in the RBC count. So once the RBC is normal, increases the O2 carrying capacity of the blood. So that is the result. Now erythropoietin is needed for conversion of stem cells in proerythroblast and further growth into mature RBC. Or simply I would like to say that this erythropoietin is stimulates the process of erythropoiesis on every stage. Every stage of the different types of immature RBC formation is stimulated by the erythropoietin hormone. So please remember erythropoietin. What is erythropoietin, where it is synthesis occur, where it is synthesized, and all the things, because it's an important viva question. Now factors affecting erythropoiesis. Maturation factors: vitamin B12, intrinsic factor, and folic acid. Vitamin B12, which is also known as extrinsic factor, as this factor is must be present on our food, in our food. Function: synthesis of DNA. Source: meat, milk, and liver. So majorly this vitamin B12 is from the animal source. So the person who are a pure vegetarian having more chances from the suffering of vitamin B12 deficiency. Daily requirement 1 to 3 microgram. Deficiency causes the failure of multiplication and maturation. So this type of condition is known as megaloblastic anemia. So the anemia due to deficiency of vitamin B12 is known as megaloblastic anemia. Now the intrinsic factor: source, parietal cells of the stomach. Function: absorption of vitamin B12, and its deficiency causes pernicious anemia. So you must need to remember what is pernicious anemia and what is megaloblastic anemia, and what is the difference between these two types of anemia. So first of all, we already seen the megaloblastic anemia, which is due to deficiency of vitamin B12, and pernicious and the pernicious anemia, which is due to deficiency of intrinsic factor. This intrinsic factor is synthesis from the parietal cells of the stomach, and its function absorption of vitamin B12. So if the intrinsic factor is not present, then the absorption of vitamin B12 is not occurring inside our gastrointestinal tract. Folic acid: source, vegetable, fruits, and meat. Daily requirement 50 microgram. Function: synthesis of DNA. So deficiency causes maturation failure anemia or folic acid deficiency anemia. So this folic acid deficiency anemia or maturation failure anemia due to deficiency of folic acid in our diet. Factors affecting erythropoiesis continuing. Iron. Function: synthesis of heme part of hemoglobin. So hemoglobin consists of heme plus globin, and in the synthesis or during the synthesis of heme part of the hemoglobin, the iron is necessary. Source: meat, peas, leafy vegetables, and jaggery. Daily requirement 1 to 2 microgram. Daily requirement 1 to 2 milligram. The body stores iron in hemoglobin, which is around 65%, the liver, spleen, and bone marrow. Intracellular iron is stored as ferritin and hemosiderin. So here the two different types of iron storage chemicals, which is or substances, which is one is the ferritin and second one is the hemosiderin. Deficiency of iron causes microcytic hypochromic anemia, or simply it known as iron deficiency anemia, due to deficiency of iron in their diet. So we already seen the vitamin B12 deficiency anemia, folic acid deficiency anemia, and iron deficiency anemia. These three anemia collectively known as nutritional anemia, because it is a anemia where the cause is due to lack of nutritious substances present in the diet. So vitamin B12 deficiency, which is known as megaloblastic anemia. Next is the folic acid deficiency anemia, and here is the iron deficiency anemia. So all these three collectively known as nutritional anemia. Other factors: copper, magnesium, cobalt act as a catalyst, calcium, other hormone thyroxine, ACTH, adrenaline, and testosterone. High protein diet, which helps in the synthesis of globin, vitamin B6 for enzymatic action, and vitamin C to keep the iron in ferrous state. Bile salts for absorption of metals and fat soluble substances. Fate of RBC: the lifespan of RBC is 120 days. Old erythrocytes becomes rigid and fragile. Hemoglobin begins to degenerate. Engulfed by macrophages, heme and globin are separated. Iron is salvaged for reuse. Globin is reutilized for the formation of polypeptides. Pyrrole rings of hemoglobin form yellow bile pigments, bilirubin and biliverdin. So the normal count and variations. Normally, in males 5 to 6 million RBC per cubic mm of the blood. In females 4.5 to 5.5 million RBC per cubic mm of the blood. In fetus 6 to 7 million RBC per cubic mm of the blood. In infants 7 million RBC per cubic mm of the blood. So this is the normal RBC count which you must need to remember. Variations: polycythemia, increased count. Whenever the RBC count is increased, the condition is known as polycythemia, and whenever the RBC count is decreased, the condition is known as oligocythemia. But here we are more use the word anemia. Here we are more using the word anemia to denote decrease in RBC count. Variations: physiological variations and pathological variations. The diurnal variation more in the evening than in the morning. Exercise having a stimulating effect over the larger amount of RBC released during the exercise. High altitude also increase the RBC count. Temperature, increase in the temperature having a positive effect on the number of RBCs present inside our blood circulation. Adrenaline also having a positively effect or increase in the RBC count. Excitement also somewhat increase in the RBC count. Males having a double plus or positive effect on the RBC count, but here the age is inversely proportional to the RBC count. So here as the age increases, the RBC count becomes decreases. As compared to old age, the younger ones having a higher RBCs in their blood circulation. Sex more in males due to testosterone. So here if you notice our previous slide, that the normal value of RBC count in males is higher than that of the females because it is due to the testosterone hormone, pathological cardiorespiratory disease, and hypoxia. Abnormal shape and size of RBC. Anisocytosis, which means change in the size. So microcyte, smaller RBC, and macrocytes are the larger RBC. Or simply, the smaller RBCs are known as microcytes, where the larger RBCs are known as macrocytes. Poikilocytosis, change in the shape. Whenever there is a change in the shape, the condition is known as poikilocytosis. Spherical in spherocytosis, and the sickle shape in sickle cell anemia. I hope you understood about the red blood cell.